All right, everybody, going through chapter 8, notes on blood and blood spatter. In this chapter, we're going to talk about what the composition of blood is, what makes up the blood, what's the function or the job of the blood cells. We will be able to identify the blood type of a blood sample. Conducting a blood spatter analysis, we likely won't be able to get to since we're doing things online. We will look at some wounds and describe the nature of the weapon and find and process blood evidence. I'm talking about the composition of blood. So what makes up blood? So the blood that we have in our body contains, is what we would call whole blood. Okay, whole blood carries the cells and the plasma. Okay, plasma is fluid with hormones. Okay, it has clotting factors and nutrients. Clotting factors are the things that allow our blood to stop bleeding. So if you get a cut on your arm, eventually it'll stop bleeding and it'll sort of scab over. That process is due to clotting factors. Okay? And then we have red blood cells. Scientific name for red blood cells is the erythrocyte. They contain hemoglobin. Hemoglobin is a protein that carries oxygen to the body's cells, and it also carries carbon dioxide away. The bright red color that you see in some blood is because that blood is full of hemoglobin. It's full of oxygen. When you see blood that's more the darker side, the darker blue or darker purple kind of color, that is blood that is full of carbon dioxide. So red blood cells are the most abundant cells in our body, and they are produced in our bone marrow. White blood cells, or leukocytes, are known for fighting disease and foreign invaders. Okay, they contain cell nuclei and produce antibodies. Okay, these antibodies are proteins that white blood cells secrete, and they attach to antigens to destroy them. So anything bad that comes into your body, these white blood cells are going to attack them and fight them off. Platelets, or thrombocytes, they aid in blood clotting, and they help to repair damaged blood cells. They clot together in a process called coagulation. That's what seals a wound and creates that scab. And they are clotting factors that are carried in the plasma. Plasma is a yellowish liquid portion of the blood, contains electrolytes, nutrients, and vitamins, hormones, and clotting factors. Oftentimes you might hear of people donating plasma and receiving some sort of a monetary benefit from it through like BioLife Plasma Services. That is something that is available in our area. So some facts about blood. The average adult body has about 5 liters of blood. So that means that 7 to 8% of your body weight is blood. Okay, there are about 1 billion red blood cells in every 2 to 3 drops of blood. For every 600 red blood cells, there are about 40 platelets and only 1 white blood cell. Blood typing. Okay, so your blood type is established before you are born. There are certain genes that you inherit from your parents, okay? So one of them comes from mom and one of them comes from dad, and these genes determine what your blood type is by causing certain proteins called agglutinogens to exist on the surface of all of your red blood cells. Okay, there are 3 alleles that we use for blood type: A, B, and O. And so A, B, and O make a variety of different combinations of different blood types. So you can be AA or AO, which means you're type A, BB or BO, which means you're type B, OO, which is type O, or AB, which is type AB. Okay, we're going to talk more about that as we get going. So if we were to look here at this diagram, it says the percentage of the population that has these certain blood types. Okay, about 42% of the population is type A, 12% of the population is type B, 3% of the population is type AB, and 43% is type O. Okay, blood typing is a quick and very less expensive than DNA profiling. So if you find blood at a crime scene, they are going to look at typing the blood, so figuring out, okay, what blood type is this and what's the blood type of our suspect. If they don't match, it's a cheaper and faster test to figure out that that's not correct, okay, and that they need to be looking for somebody else. So the RH factor. This RH factor is where you may hear of people say, well, I'm A positive or I'm O negative. That positive or negative is referring to this RH factor. Okay, while studying rhesus monkeys, okay, which are often studied in science because they are similar to humans, there was a certain blood protein that was discovered. The presence of this protein, or really the lack of it, is referred to as the RH. Okay, so if it contains the blood, it's called RH positive, or if it contains the protein, it's called RH positive. If it does not, it's called RH negative. So me personally, I know that I am an A positive blood. Okay, that means that I have type A blood and I also have this RH positive protein in my blood. So antigens, these are foreign molecules or cells that react to antibodies, and then enzymes. Enzymes are proteins that catalyze different biochemical reactions, meaning they make chemical reactions happen and they happen to speed them up. Given the frequency of different genes within a population, it's quite possible to determine the probability that a particular blood type will appear in a particular population. Okay? You are able to determine this by multiplying individual probabilities and identifying the additional proteins in the blood sample. So if we were to look at the distribution of blood in a sample size, okay, O positive, about 1 in every 3 people is O positive. O negative, 1 person in every 15, and so on. So you can see what that situation looks like. Okay. So blood is evidence. So going back to the first unit of forensics, we talked about class evidence, individual evidence. So blood typing is class evidence because it does not lead you directly to the person who committed the crime. It can tell you a group of people who have that same blood type. DNA profiling, however, is individual evidence because it will direct you to that specific person who committed the crime. A blood spatter pattern is used to identify or verify an account by a witness or a suspect. Helps us find the origin of the blood, the angle, and the velocity or speed of the impact, and the type of weapon that is used. Okay. If we are looking to determine whether it is animal blood that we found at a crime scene or human blood found at the crime scene, they will use this test called the precipitin test. Okay. So blood is injected into a rabbit, antibodies are formed, rabbit's blood is extracted as an antiserum, and then the antiserum is that's what's placed on the sample blood. Okay, if it reacts, it means that there's human blood present. Okay, it's very sensitive. It does not require us a lot of blood, so that's beneficial. So if you look at animal blood here, for example, frog blood, they have larger nucleic red blood cells. So you can see in the center those large nuclei. Human blood, on the other hand, does not have a nucleus. Okay, you can see the human blood cells, the red blood cells, are very numerous. So there's so many of them in comparison to the white blood cells. Okay, white blood cells are larger and platelets are very tiny cellular fragments. So blood as evidence, okay, you can take a blood sample to determine the blood type and the DNA. Blood droplets will give clues to the location of a crime, movement of a victim, and type of weapon. We will address this later, probably next week. Blood spatter can be analyzed to determine patterns, and so that will help give the investigator some clues as to how a crime might have happened. So in 1939, the meaning of a blood spatter pattern was first analyzed. When a wound is inflicted, a blood spatter pattern may be created. Okay, it takes a group of blood stains to make a pattern, and it can help recreate or reconstruct the events surrounding a shooting, stabbing, or a beating. Okay, when they analyze it, they are able to determine the direction that the blood has traveled, the angle of impact, the point of origin, and the velocity of the blood. as well as the manner of death. Okay? When blood falls at different heights or at a certain speed, it can cause natural cohesiveness. Cohesiveness means it's trying to stay together, and that can create a satellite droplet. So when you look at this image on the top right-hand portion of the slide, the satellite, you can notice, is outside of the actual blood droplet. Then we're talking here about a spiking pattern. That is where it is still connected to the original blood droplet, but it comes out to the side of it, kind of spikes out. So these different types of spatter patterns, okay, that you see up here on the screen, 1, 2, 3, 4, 5, 6, can be described by each of these, okay? And so we will get further into this, and you will see explanation of this also in your textbook in Chapter 8 when we go through there next week. Okay, patterns can help investigators determine the type of weapon that's used. Okay, so if you were to look at this, the image on the left is the kind of spatter pattern that you would see that's produced by a gunshot. Okay, you can see it's splattered all over, very, very small pieces because of the high velocity. The speed at which a bullet would hit a human being or an animal would be much greater than a hammer blow. Okay, so the one on the right is produced by a hammer blow. So that could be a hammer, it could be a baseball bat, anything that is heavy that you would hit someone with. And you can see there are some splattering, but it's not as much because the force and the speed is much different in this situation. Okay, looking at directionality, okay, the shape of an individual blood drop provides clues to the direction of where it originated from. Okay, this is going to talk about lines of convergence, and we will get into this next week, and we will address that. So again, lines of convergence, if you have all of these blood drops, you are going to draw a straight line through the center, and where those lines for all of those blood drops intersect on that X and Y axis, that general area is where we can assume the victim was, the wound was inflicted on the victim. And so that is going to help recreate some of those crime scenes and what has happened with them. Okay, so crime scene investigation of blood, looking for blood evidence if there's any possible when they arrive at the crime scene. If there is, they need to determine, is it human blood or is it animal blood? What blood type is it? And then interpret those findings to figure out if it's beneficial information to them and if what steps they need to follow in finding that suspect. Okay, summary. This is just identifying all of the stuff that's been covered in the notes. There is going to be an exit ticket that I'm going to have you fill out on Google Classroom, so please make sure you do that to get credit for this work.